Hello, welcome to Bio 23 Infectious Disease, Chapter 9, where we're going to talk about bacterial genetics and give a general introduction to genetics as well. Remember that all of life on Earth inherits its information from parent to offspring as DNA. DNA stands for deoxyribonucleic acid, and it's similar to the molecular structure of RNA. RNA stands for ribonucleic acid. Both of these are very similar in structure and composition. Both DNA and RNA are made of nucleotides. Nucleotides are comprised of three individual units. Here we have the basic deoxyribose sugar. Deoxyribose sugar is found in DNA, hence the name deoxyribonucleic acid, phosphate, and a nitrogenous base. All nucleotides contain these first two components, the sugar and the phosphate. What makes the different nucleotides different from each other is the nitrogenous base. In DNA, it's either A, T, C, or G, otherwise known as adenine, thymine, cytosine, and guanine. DNA is made of deoxyribose sugar, whereas RNA is just made of ribose sugar. Ribose sugar has an added oxygen atom bound here. In addition, another difference between DNA and RNA nucleotides is that while DNA has the A, T, C, G nucleotides, RNA has A, U, C, G. So instead of thymine, RNA molecules use uracil. Here's the structure of those nitrogenous bases. We have guanine and adenine, and these are classified as purines, and cytosine and thymine, and these are classified as pyrimidines. You can see the pyrimidine structure just has a single ring, whereas the purine structure has two rings. Uracil is also considered a pyrimidine because it's very similar to the thymine structure. When these nucleotides come together, they form a nucleic acid strand. And in DNA, there's two nucleic acid strands that come together. Here is the nucleotides as seen joining together to form the nucleic acid strands. So here we have the deoxyribose sugar marked as D, the phosphate marked as P in orange, and the nitrogenous bases located in the middle of the DNA molecule. As you can see, the backbone of the DNA molecule, what holds the nucleic acid strands together, is this phosphate sugar backbone. Phosphate sugar, phosphate sugar, phosphate sugar. So we refer to this as the phosphate sugar backbone. In the middle of the DNA structure with the two nucleic acid strands, we have our nitrogen bases. The nitrogen bases are what hold the two strands together. The phosphate sugar backbone is held together by covalent bonds, which are extremely strong bonds. The bonds that hold the two nucleic acid strands together are hydrogen bonds, and these are much weaker and easier to break. As you can see from the picture here, G pairs with C and A pairs with T. I tell my students they can remember this because A and T are made of lines. They are letters that you can draw with lines, whereas C and G are curvy letters, and so you can remember that they bind together that way. Another thing of note in the nucleic acid structure is that this whole structural complex is in a coil. As you can see, it's twisted. This is what's referred to as the double helix. Double because there are two nucleic acid strands, and helix because it's in a coil. Your book and many other books describe this as a ladder that has been twisted. So the sides of the ladder are your phosphate sugar backbone, and the rungs of the ladder or what you would normally step on as you're climbing the ladder, the middle part, is made of nitrogen bases. Now remember that this DNA molecule is what's required and necessary for the information for life. So this is what carries information to make living systems. In order for a cell to replicate and to generate daughter cells, that cell must first copy its DNA molecule. That is the most important thing it shares with its next generation. This process of copying DNA is known as DNA replication. The way DNA replication works is first, those two strands of the DNA molecule are pulled apart. Remember, this is fairly easy for enzymes in the cell to do because you're breaking hydrogen bonds in this process. When those hydrogen bonds are broken apart, this is what's referred to as unzipping, because it's like unzipping a zipper. Unzipping begins at a specific sequence in the DNA molecule known as the origin of replication. So this is where DNA replication, copying DNA, begins. When the DNA molecule is split apart like this, it reveals what's on the inside of that double helix. What's inside that double helix is the nitrogenous bases, and that's what makes every individual unique. Every cell on the planet has the phosphate sugar backbone for its DNA. That's not different at all. What is different from one cell to another is the nitrogen base content on the inside. So by opening up the double helix and unzipping it, you're revealing the information that makes the cell unique. This is the information that needs to be copied to form the next generation of cells. There is a single enzyme that's primarily responsible for DNA replication. There's in fact 6 or 7 enzymes involved in this whole process, but the most important enzyme involved in DNA replication is the enzyme known as DNA polymerase. It's called DNA polymerase because it is an enzyme, so the word ends in ase, that's involved in making DNA molecules or polymerizing DNA. DNA replication is also referred to as semi-conservative, and the reason why we call it semi-conservative DNA replication is because the way that DNA is replicated is, first, we said that it unzips, so the two parent old strands become separated from each other, and then DNA polymerase adds a single nucleotide at a time to generate a brand new strand from scratch. What this results in are 2 double helices, or 2 nucleic acid DNA molecules that are meant for the 2 daughter cells, and they are genetically identical. And as you can see, each one of them contains an old strand, marked here in purple, and a completely new strand here in orange. So semi-conservative DNA replication means half of the DNA is conserved or remains intact. That would be the purple strand, whereas the other half of new DNA molecules are totally new. Now that is, in very basic terms, how DNA is replicated. Remember, DNA is only replicated for cell division. It's only replicated when the cell knows that it needs to divide. There are many cells in your body, as an adult, where you will not need to grow and divide those cells anymore. For instance, the cells in your brain, your heart, your kidneys are done growing. And since they will never divide again, they will never go through DNA replication again. However, the next 2 steps that we're going to talk about, transcription and translation, are happening in all of your cells all the time. Transcription is taking the information that's found in your DNA molecules and using that information to make RNA. Your DNA molecule is essentially like a cookbook. It's kind of like the How to Cook Everything for This Human Being cookbook. But while every cell needs to have the entire cookbook, not every cell is using all the recipes all the time. So for instance, your heart muscle cells don't need to filter blood like your kidneys do. And your kidneys don't need to pump rhythmic muscular contractions like your heart does. So your heart uses some of the recipes from the cookbook, and your kidney uses other recipes from the cookbook. They all have exactly the same cookbook, but they use different recipes. It's kind of like making breakfast versus making dinner. You wouldn't be making pie for breakfast necessarily. And so you choose the recipes that you want for the time and place and tissue type that you'd like. When you choose a recipe, this is transcription. So what the cell does is it takes this cookbook, this very important original piece of information, DNA, and it makes a copy of the recipe that it will take into the kitchen. Rather than take the really important cookbook into the kitchen, where it could get damaged or destroyed, the cell makes a copy of the recipe that it's going to use, which we call RNA, and then it makes the proteins, the recipe itself, from just the RNA molecule that it copied. This whole process of transcription and translation is what we refer to as gene expression, because the piece of DNA that we're copying, the recipe, that is your gene. And when we say a gene is expressed, we're saying that the gene is being used. So when we use the gene, we're copying it into RNA in transcription, and then we're converting that information to a protein, or making the recipe, in translation. Let's first talk about transcription. Transcription is very similar to DNA replication, where the first step is to pull the molecule of DNA apart at its hydrogen bonds in the middle to reveal the important information inside. Then an enzyme comes and adds nucleotides one at a time to generate a product. The difference is, though, is that here the product that you're making is RNA. And so the nucleotides that are being added are RNA nucleotides. The other main difference that we see here is that the enzyme responsible for this is RNA polymerase, not DNA polymerase, because you are making RNA in this case, so you use a slightly different enzyme. Finally, the other thing that you'll notice is that unlike DNA replication, RNA transcription is only making a single strand. You are not copying the opposite strand of the DNA molecule. Finally, another difference between transcription and replication is that transcription starts at a different sequence, and it actually ends as well. Transcription, remember, is just copying a recipe or a gene. And so you're just copying a section or a portion of the DNA, not the entire DNA molecule like in DNA replication. So you need to have a sequence where you start transcription as well as a sequence where you end it. Transcription starts at a sequence called the promoter sequence. It is different than the origin of replication. Transcription ends at a sequence known as the termination sequence. These are very specific terms and don't go by any other name. So RNA polymerase will grab onto the promoter sequence, open up the DNA strand to reveal the information inside, add one nucleotide at a time to generate the RNA molecule, and then when it hits the next sequence, the termination sequence, it will release the RNA molecule and the DNA strand will close behind it. That's transcription, but only half of gene expression. Remember, we've just copied the recipe. We haven't actually taken it to the kitchen and made the recipe. That process is translation, and that's what we'll be talking about in the next section.